All right, so we're going to finish up talking about the eyeball and the gross anatomy before we get into how the eye actually processes the information. So, the innermost layer of the eye is going to be where you have the neurological components, the receptors, the photoreceptors. So, the posterior region of the eye is going to have the inner layer known as the retina. And that is originally an outpocketing of the brain, which, if you remember from class, I told you there was four components to the diencephalon, and I asked that even on an exam, and I don't think any of you put down retina, and that is the fourth component. So it's always the one that's forgotten. So that's why I wanted to make sure that I hammered on that. So there are four components: the hypothalamus, the epithalamus, and the thalamus, and then there's also the retina, which is originally part of the brain. So there is the neurological component. There's receptors and neurons that will be found there, and that is how you perceive vision. You see light beams, and you create images from that. So it's a delicate two-layered membrane. You have the outer pigmented layer, which is going to be, of course, colored, and that is going to be a single cell thick lining. So you have one layer of cells, and this absorbs light and prevents it from scattering. So when it is pigmented, it means it absorbs light, and it makes sure that it phagocytizes the cell fragments so that they do not disrupt the image that is coming into the eye. This also will store vitamin A, and vitamin A is vital to vision. So if you do not have vitamin A in your diet, it can affect and distort your visual fields. So vitamin A can lead to night blindness and other issues. So make sure that you're getting your fruits and vegetables in that contain vitamin A. So the inner layer is going to be your neural layer, and this is going to be transparent, okay? And it's obviously made up of neurons. It's three main types of neurons. You have the photoreceptors, which are your rods and your cones. Then you also have bipolar cells and ganglion cells. So we're going to get involved and in depth with what each one of those means. So the signals will spread from photoreceptors to bipolar cells to ganglion cells. And the ganglion cell axons will exit the eye as the optic nerve. So you collect the information from the photoreceptors, which passes the information to the bipolar cells. And bipolar cells means the cell body is in the middle of the axon. So you have an axon on both ends. And then it'll pass that information on to the ganglion cells, which is the neuron that will leave the eye, and it will actually go into the occipital lobe of the brain, which is in the posterior of the cerebral cortex. Now, where it leaves the eye is known as the optic disc. This is your blind spot. So where your nerves leave, you don't have any photoreceptors, okay, because you have to have neurons traveling somewhere. And so there's a small area in the eye that is where all of those neurons will leave, and lacks photoreceptors. You can't collect any visual data. You don't absorb that light here. So it just like I said, it's a blind spot. You don't have any ability to process that information. In the rest of the retina, you have 250,000,000 photoreceptors of two different types. You have rods and you have cones. Okay, so we're going to discuss each of those very in depth. So you're going to really need to know the difference between rods and cones, okay? And those are going to be your photoreceptor types. And again, you need to know what a photoreceptor is, because that is the only receptor in the body that perceives light. It helps you create images in your brain, so you can see, you can perceive somebody's face, you can perceive the colors around you because of the rods and the cones. Okay, so you need to know that. If I ask you a question that says, okay, name three special senses and what receptor is found in each, you can say, well, there's vision, which is a special sense, and you sense that by photoreceptors, which are your rods and cones. So if you get in depth and say rods and cones for vision, okay, that works. Then you also would need to tell me something about taste or smell, and that it's a chemoreceptor, or equilibrium and hearing, which is mechanoreceptors. So here is an image of the ganglion cells. So your photoreceptors are back here. Then you have the bipolar cells, which are going to take that information and bring it to the ganglion cells. Those ganglion cells are then going to travel all the way to the blind spot. It's not directly posterior, it's actually it's off center. And so it's going to be where you exit all of those neurons and have the optic central arteries and vein of the retina coming into the center of the eye. Okay, so not into the choroid and the vascular layer. Those actually go into the internal eye, and we'll discuss that again later. So this would be the blind spot. Again, you have no photoreceptors right here. Okay, that's where all the neurons exit and those blood vessels come into and out of the eye. Okay, so this is a better picture that shows you the rods and the cones. And so the cones actually look like they have a little cone on the end, and then the rods look like they have a rod. And they connect to these bipolar cells. So here's the cell body. It has one axon here, an axon there. So it's in the center of the axon, and you have the dendrites, and then the axon terminal over here communicates that information to the ganglion cells, which will then leave as the optic nerve. Okay, so the light will come in from here, bypass the ganglion cells, bypass the bipolar cells, and will be stimulating the rods and the cones. So light will pass through those cells, those neurons, because those neurons do not have the receptor available to understand what or perceive light. They don't have that communication capacity. So the only cells here that will actually interpret vision and interpret light waves are going to be those rods and those cones. So light passes by the cell bodies that cannot detect the wavelengths, and it will go just to the cells that can, and then you send a signal back the other way towards the optic disc where the blind spot is, and the optic nerve exits the eyeball. Okay, so here's an image of an actual eye. And so again, light will pass from here down to the rods and the cones where you will perceive light. If anything gets by there, the choroid layer, which is pigmented, will absorb the wavelength so it does not reflect backwards and cause a double stimulation of the rods and cones, which will lead to blurry vision. Here are the nuclei of the rods and the cones. So the receptive region is right here. Okay. And there's the pigmented region, which is going to absorb light, and more light would be absorbed here if it does get that far. Okay, and then you have the nuclei of the bipolar cells, so you have the axons of the rods and cones and the bipolar cells, and then you have axons here that are going to travel out. Here are the ganglion cell nuclei, and then the axons are all going to travel in this region as well, which will lead to the optic disc. So let's discuss the difference between rods and cones. Well, rods are going to be more frequently found in the eye. They're going to perceive dim light and peripheral vision. Okay, so they are going to be much more abundant, especially in the periphery of the eye. So you're going to find that they are more sensitive to light than cones. Obviously, they're much more numerous, but they do not detect color vision or sharp images. So these would be like your black and white TVs. Okay, you get the gray images. It's slightly blurry, but you can still, little amounts of light can actually activate these rods. So they're most numerous at the periphery, and these will be utilized a lot more in the dark. So if you try to find your way in when it is very dark, so say you get up at 11 o'clock at night and you don't turn any lights on and you're trying to find your way to the bathroom, you can kind of see what's around you, but it's blurry. You have no real color associated with it. It's all in gray tones because your cones aren't activated. It's only your rods, and that will allow you to maneuver and get around, but it's not going to give you the HD image. Cones are going to be the opposite. So these vision receptors are for bright light. So this gives you your high resolution or your HD color vision. So that's your 4K TV. It's got everything that you want. You can see in the most detailed aspects, and they're going to be found mostly in the macula lutea, which is exactly at the posterior pole. That is going to be where you have your fovea centralis. It's a tiny pit in the center of the macula with all the cones. That's where you get your best image. So you're trying to, when you are focusing on something, direct as much light as possible to that fovea centralis so you get the best, clearest, brightest image for your body to work with. Okay, so that's mostly cones. There's few rods present there where you get your HD image. Now, the blood supply here is going to be the choroid that supplies the outer third, which would be for the photoreceptors, and then you have the central artery and vein of the retina that supplies the inner 2/3. So you have two different ways to get light into to get nutrients to the neurons, and so you have a lot of these receptors that are going to be utilizing ATP. It's going to be utilizing energy, glucose, oxygen on a very regular basis because they're constantly being activated by the light that we are encountering in the environment. Okay, so the central artery and vein will enter and exit the eye in the center of the optic nerve, which is where you have your blind spot. And you can actually see the blood vessels in a living person. So if you are doing an eye exam and you shine a light into the pupil and you get some magnification, you can actually see the arteries and the veins in the eye. And so you can do those tests to see if there is some damage to the eyes with something like diabetes or strokes. And that's something that an ophthalmologist would use. So here is your macula lutea, and so that's going to be your dense spot that's exactly at the posterior pole where you're going to focus as much light as you possibly can, and that's where you're going to develop your HD image. Okay, but if you notice that just to the side here, this is going to be your optic disc. So that's where you have no photoreceptors. Okay, no rods, no cones. All it is going to be the artery, vein, and the neurons, axons that are leaving. So the ganglion axons that are leaving the eye are all present here. And so you can see all these blood vessels, the central artery and vein that emerge from the optic disc and spread throughout the internal eye. So these are visible when you're looking inside a person's eye, and it's something that you'd have to do a clinical examination to get more information, because we're not going to get in depth on that. But if they are engorged or inflamed, there's usually something impinging on those blood vessels. Okay. So what are the fluids that are within the eye? I already teased that with the two humors. Okay, so the internal chambers, which are separated into anterior and posterior segments, will have two different types of fluid, and they'll have different purposes, they'll have different makeups, and they'll also have different origins. So the lens and the ciliary zonule separate the eye into two segments. It's the anterior and the posterior segment. Okay, so the anterior segment is going to have the aqueous humor, whereas the posterior segment is going to contain what's known as the vitreous humor. Okay, so the posterior segment contains vitreous humor that is going to help transmit light, because essentially that's what the purpose is going to be. You want to transmit light into the retina so that you can create vision. Now it's also going to support the posterior surface of the lens, okay, maintaining its hold where it needs to be. So you want to maintain the lens within a certain area so that it doesn't move. Otherwise, it's going to bend and reflect light to a different portion. We want it always to bend and bend light so that it lands on the focal point in the back of the eye. It also will hold the neural layer of the retina firmly against the pigmented layer, so if you lose some of that vitreous humor, that retina can detach, and then you will not get clear images. You actually might even lose your vision, because then you're losing the connection to your blood supply. It contributes to the intraocular pressure, and this actually forms in the embryo, so it lasts a lifetime. So when you create this vitreous humor, it's going to stay in the posterior segment of the eye for your entire life. Okay, and the intraocular pressure, you want to maintain at a certain point. If it gets too much, it can do damage. If it gets too little, it could lead to retinal detachment, which again is potentially going to ruin vision and cause deterioration of the photoreceptors. Okay. The anterior segment, on the other hand, is going to be made up of aqueous humor, and that is going to be separated into an anterior chamber and a posterior chamber. So you have the anterior and posterior segments, which are going to be separated by the lens, which contain two separate humors. And then the anterior segment is divided into the anterior-posterior chamber, which is separated by the iris. And the anterior chamber is going to be where you drain the aqueous humor, where the posterior chamber is where you are going to create the aqueous humor. Okay. So the anterior segment contains aqueous humor that is a plasma-like fluid continuously formed by capillaries of the ciliary process. So what that means is you're taking blood plasma and you are taking certain components of it and you are taking them out of the capillaries so that they can flow through the anterior segment of the anterior chamber, or the anterior chamber of the anterior segment. So it's going to drain via the scleral venous sinus, which is also known as the canal of Schlemm. So yes, there are two different names, and you need to know them both, because some guy named Schlemm decided he wanted a canal named after him, and anatomists now are utilizing these other forms of naming so that it makes more sense. Okay, so scleral venous sinus means that it's from the sclera, it's a venous sinus. So the canal of Schlemm is being phased out essentially, but a lot of people still utilize that term, especially in clinical settings. So you're going to need to know both, so that if somebody says canal of Schlemm, you understand what they're saying. Okay, so this humor is going to supply nutrients, oxygen to the lens, the cornea, and then also somewhat to the retina. It removes waste because it's a constant flow. So you can't have blood in your aqueous humor or in the anterior segment because, well, I mean, blood vessels and red blood cells, white blood cells, large proteins will all distort vision. So you need to only take what you absolutely need from that blood plasma that will not reflect and refract light and allow it to flow. Okay, and then you also are able to drain different wastes and toxins that are no longer necessary. If those build up, that can cloud vision and lead to some damage. And actually, glaucoma is a blocked drainage of aqueous humor, so that increases the intraocular pressure, causes compression on the retina and the optic nerve that can eventually lead to blindness. And there's two different forms. There's opened and closed glaucoma, and I believe opened is the extremely painful one. One is very painful, one is not painful, or not as painful. And so you can actually have two different types of pressures that are formed from glaucoma. So you want to treat that before it causes so much pressure that it actually kills off some of those photoreceptors, which would lead to blindness. Okay, so we've talked about several of the different components. So here is the lens, the suspensory ligaments, and the ciliary bodies. So this line here is separating the anterior and the posterior segments. So here is the posterior segment with the vitreous humor, which is lasting a lifetime. It's created in the embryo. Back here is the optic disc with the optic nerve, the central artery and vein, and there is the fovea centralis with the macula lutea, or that's the focal point. Okay, so it's directly posterior to the lens and the pupil. Okay, here's the pupil, it's the open space. Here's the iris, okay, and so this line here separates the anterior and the posterior chambers of the anterior segment. So these ciliary bodies are going to secrete or filter the blood plasma to create that aqueous humor, which is then passed up into the pupil, which will then exit the pupil and come down and be drained by the scleral venous sinus, also known as the canal of Schlemm. So the fluid will travel from the ciliary bodies up, out the pupil, down, and then be drained. Okay, so that you continuously are creating new aqueous humors that will be flowing out. And if this is closed off, that can lead to glaucoma, because now you increase pressure because you're not stopping the production of aqueous humors that pushes the lens, pushes the posterior segment, the vitreous humor, and it creates pressure on the photoreceptors that are very sensitive that could lead to blindness. Here's the cornea. So you can see how much that would bend light, and that's actually the part of the eye that bends light the most. The lens will fine-tune that so that you direct light to the fovea centralis. Okay. So the aqueous humor forms by filtration from capillaries in the ciliary processes. It flows out, up to the pupil, out of the pupil, into the portion between the pupil, the iris, and the cornea. Now you have all this clear fluid that flows from the posterior chamber to the anterior chamber. Some will flow through the vitreous humor, but it's pretty minimal because this is going to be separated, and also it's going to be lasting your entire lifetime. But you're going to want to really clear out that anterior chamber, and then it's drained through the scleral venous sinus, where it is reabsorbed by blood vessels. So you can see those blood vessels, and now it's going to be transported back to the heart for recirculation. So what is the lens? Well, the lens is a biconcave, transparent, flexible, and avascular structure. So there's no blood vessels to it, which is how it remains clear. It's flexible so that you can moderate the amount of light that is bent and how much where it's focused. Okay, so that you can change how you are focusing from something very close to something very far away. Transparent, so light easily passes through it. And it's biconvex, so it's actually bent twice when you go through the lens. Once as light enters the lens, and once as light exits the lens. So it changes shape to precisely focus light on the retina, and specifically on the fovea centralis. Okay, so lens will become more dense, convex, and less elastic with age, which means that it is less capable of focusing as you age. And so this can lead to what's known as presbyopia, which is, in other words, as my instructors used to call it, old eye. And so that's when you use the glasses known as cheaters, so that you can focus on images that are close to your face. So people who have presbyopia have to hold objects far away from them so that they can get a clearer image. They don't have the ability to focus on something too close to their face. Cataracts is going to be when you have clouding of the lens, so you're no longer able to get rid of the waste products within the lens that can lead to damage of the lens and clouding. Okay, so it's a consequence of aging. Diabetes mellitus, people who suffer from diabetes will actually have a lot of issues with this. If you have heavy smokers, will also develop cataracts. And frequent exposure to intense sunlight. So this is somebody who sits out and stares at the sun, not necessarily somebody who is out in the sun all the time, but that could lead to some clouding as a mechanism for protection. So that would be what cataracts looks like. Look at all the pretty colors. So as you shine a light into the lens, it's clouded. Those light waves are going to be refracted, and it leads to very cloudy images. So this person might have a lot of blurriness, and it's very difficult for them to read. And that's typically not a younger individual. They typically don't have problems with that. Okay. So how do you see? Okay. Let's talk a little bit about how you actually perceive and sense light waves. Because eyes respond to visible light. So a small portion of the electromagnetic spectrum is seen by the human. Only a very small portion. We don't see things like X-rays or microwaves. We see different colors, and actually we only are able to absorb 3 different colors, but the combinations allow us to see a variety of colors. So the wavelengths are going to be 400 to 700 nanometers. Again, that's a very tiny portion of the amount of light waves that are present in the world. So light is there are packets of energy known as photons or quanta. So we're not going to get into physics and quantum physics, but they travel in a wave-like fashion at very, very, very high speeds, much faster than sound waves. That's why you see lightning before you hear thunder. So the color of light objects reflect determines the color the eye perceives. So whatever is absorbed is not something you see. So black colors will actually absorb all light waves, so you don't actually get a reflection of the light. White actually, on the other hand, is going to reflect all colors. Okay, so that's why wearing black shirts on a very sunny, hot day is bad, because it absorbs all the sunlight and absorbs all that heat, whereas white will bounce all the heat waves off of it. Okay, so here's the electromagnetic spectrum. So we see blue, green, and red. Okay, so those are the cones that you have in your eyes. And so when you can see blue images when they absorb that blue light. You can see green when green does, and same with red. And as you combine those colors and those cones in certain patterns, then you're going to get different color perceptions. So that's how you see yellows and oranges and et cetera, et cetera. Okay, and then you also have like the gray tones with the rods that are stimulated. And look at how small of a portion of the wavelengths that we can see. We don't see UV, we don't see X-rays, gamma rays, infrared, microwaves, or radio waves. Those are all different wavelengths that we cannot perceive with our photoreceptors. Okay. And speaking of these cones, people who have color blindness may lack 1, 2, or all 3 of the different cones. Okay, so there are individuals who only lack the red cones, and they can still see in green and blue tones. They just might not be able to distinguish other colors of the spectrum. Okay, so as you combine these wavelengths, you can see different colors. So yellow here when you get the red, green, and rods all activated. Okay, red is when only the red cones are activated. Okay, green is when only the green cones and the rods will be activated, right? And then blue is only blue and potentially the rods, and as you get darker and darker, those rods are not stimulated. Okay, but you can see the rods are stimulated less, and they combine with the blue to create that blue color. And as you go down the line, these are the colors that you can see in this spectrum. Okay, so when you combine them together, you can see different colors. Okay, so refraction is a bending of light rays. Okay, so when you bend light rays, you can create different images in the eye. So it's due to a change in speed when light passes from one transparent medium to another. So that would be like glass to water. Okay, they're different mediums, so they bend light differently. Okay, it occurs when light meets the surface of a different medium at an oblique angle. Okay, and so a curved lens can refract light. So you have the cornea, and you have both sides of your lens that can refract light and bend it so that you focus that light directly on your fovea centralis. So this is a good image of seeing the straw. Okay, so you see a straw, and as you now bend to the glass, that image is distorted because it is refracted in that the lip of the glass more, and so you see the straw continue down. But as you get to the water, it refracts differently, and therefore it looks like the straw is split because it's a different medium. The light is bent differently. You get on the surface a different image than you do underneath the water. So it looks like the straw is in two different places, but it's really just the way that the light is refracted differently between the glass and between the water and the glass. So light passing through the convex lens is bent so that the rays converge at the focal point. If they do not converge at the focal point, that's when you get distorted images, or you get those black and white tones. You don't get a clear image because you're not focusing on the cones, you're focusing on a portion that is mostly rods. So the image is formed at the focal point upside down and reverse right to left. So you actually do see upside down and backwards in the eye. But your occipital lobe, your cerebral hemispheres, are going to flip that vision so you see it properly. The concave lens will diverge light, preventing it from focusing on certain images. So you can actually increase the concavity of the lens to prevent the light from focusing too much on one point. You diverge it more so that it lands on the right spot. So here's what we're going to show you. So this is going to be an image, so your point sources. So say you have this large image here. Well, light is going to reflect here, here, here. It's going to reflect all those different ways that will go through the pupil and land on the lens. So when it lands on the lens, now you get the light that is refracted and it pushes it towards the focal point. And so you have all the light rays in between here from that one point. And they're all going to converge because of the refraction of the lens into the focal point here, and then the opposite end is going to be up here. So light is going the opposite way. And as you see on the image below, the image is inverted and upside down. So what you are sensing is an upside down reversed image. When you send it to the brain, it flips that image so that you see things right side up. Okay? So I know this can be kind of a complex learning curve here because there's a lot going on in the eye. It's not an easy sense to learn about. But when we talk and more through this, you'll see what I'm talking about. And so keep going over the lecture, listen to it multiple times, and write down and draw these images so that as you continue to draw these images, as you continue to read and to hear the lecture, it's going to be easier and easier to understand. Okay, so the pathway of light entering the eye. And this is very, very important. I'm going to have you circle this. I'm going to have you highlight it. I want you to star it, to do whatever you need to do to remember this, okay? Because it will be on a test. So the pathway of light entering the eye first hits the cornea, where it is bent and refracted the most out of any portion of the eye. Then it goes through the aqueous humor, okay, which passes through the pupil. The light will hit the lens. It will then pass through the vitreous humor, and the entire neural layer of the retina is going to be exposed to that light, and it's going to be read and sensed by the photoreceptors. So most of it is going to be focused on the focal point, which is your macula lutea. But really, the entire eye is entire retinal layer is going to get some light, and that's how you get that peripheral vision. So light is refracted 3 times along the pathway. The most by the cornea, then entering the lens and leaving the lens are each going to refract the light again. So that's how you get the focus on that focal point. So the majority of the refractory power is in the cornea. I can't tell you how many times I've said that. It's probably about 10 times at this point. So if you get it wrong on the test, you're probably not listening to the lectures. Okay, the change in lens curvature allows for fine focusing, so you can actually adapt the lens to bend the light more or less to focus on the focal point. You can't do that with the retina. The cornea. Cornea is a fixed refractory power, so it does not change through time. Okay, so your eyes are best adapted for distant vision. So you can see things far off in the distance more easily because you don't have to really fine-tune that focus through to the lens nearly as much. So that's why people who are older tend to have more issues with their close vision, because you have to do more adaptation. So the far point of vision is beyond 20 feet. It's a distance beyond which no change in the lens shape is needed for focusing. So this is 20 feet for the normal eye. That's why we talk about 20/20 vision. So at 20 feet, can you see what the normal person sees at 20 feet? Can you read the same thing that a normal person can read at 20 feet? At that point, the cornea and lens focus light precisely on the retina. Okay, so it doesn't change the shape of the lens if you're looking at something 50 feet and then they move back to 70 feet. Your lens is going to stay the same. But if you change your vision focus point from 20 feet to 5 feet, then you have to adapt the lens so that you bend light differently to focus. So that's why if you're looking at somebody 20 feet away from you, or at the back of a classroom, for example, and you then will focus on your phone right in front of you, you might get a quick blurriness because your lens has to adapt. And it only gets harder as you age. Okay, so what happens is you get this image, the light will be coming into the cornea. It's bent as it comes into the cornea, then it's bent again and again as you go into and out of the lens, and you focus this inverted image on the focal point, the macula lutea in the back of the eye, not the optic disc, the macula lutea. Okay, so it's supposed to focus upside down and backwards so that you get as clear of an image as possible. So this is going to be activated in the sympathetic system. So the lens is going to flatten for that distant vision. Okay, that's the most it can flatten, so it does the least amount of change to the refraction of the light. Okay, so actually you flatten the lens by tightening the ciliary zonule. So you contract that, and then you continue to pull on the lens so that it flattens out that disc. When you relax those ciliary muscles, it's going to cause more bending in a thicker lens. Okay, so when you focus for close vision, you need to accommodate the lenses to focus on the focal point in the eye. Okay, so that's close objects, so less than 20 feet. That light will diverge as it approaches the eye. Okay, so you need to refocus that. So it requires the eye to make active adjustments using 3 processes that are activated at the same time. And again, you really want to know this. Okay, I'm going to have you star, circle, and understand what the 3 processes are. So one, you have to accommodate the lens. So you have to focus the image, the light from that image, directly on the posterior of the eyeball. Okay, so you have to eliminate a lot of the extra light that is being brought into the eye, and then the light that is coming in, you have to bend precisely so it hits the fovea centralis. So you accommodate the lens to focus, you constrict the pupils to eliminate some of the distorted light that could distract from the image and cause a blurriness. And then you also converge your eyes. So your medial rectus muscles will contract so that you get your eyes both moving medially, not a ton, but enough where you can focus closer, and you can bring the light from that close image that will come into the eye and focus directly on the fovea centralis. So you accommodate the lens, you constrict the pupils, and you converge the eyeballs. Accommodation, changing the lens shape to increase refraction. An increase in refraction means that it increases the bending of the light to focus on the focal point. So the near point of vision is going to be the closest point on which the eye can focus. Okay, and this can change throughout life. Again, we talked about presbyopia, which is the loss of accommodation over age 50. So the person in their 50s and 60s might not be able to hold a book 2 feet away from their eyes and read it. They might have to hold it at arm's length away from them so that they can read properly. When you start to see people doing that, that's when they need to go in and get what's known as those cheaters. So most people call them glasses that will help them focus on points closer And so they're only used for close vision. They're not going to be used for distant vision or far vision. Okay, the next would be constriction. So accommodation of the pupillary reflex constricts pupils to prevent more divergent light rays from entering the eye. Essentially, this would be scatter. Photo waves that are going to distort the image and create blurry words or blurry images. So if you don't constrict those pupils, again, when you're trying to read something close to you, you're not going to be able to get the proper you're not going to be able to get the proper image, so you don't see things clearly. You're not going to be able to read. You have to hold things farther away so that your lens has to distort less. Okay. Convergence is going to be medial rotation of the eyeballs towards the object being viewed. So this is the contraction of the medial rectus muscles bilaterally. Okay. So close vision requires parasympathetic activation. So the lens bulges for close vision, so it distorts and refracts the light more so that it focuses on that fovea centralis. You contract the ciliary muscle, loosening the ciliary zonule, and allows the lens to bulge. So some of the different problems of refraction are going to be, one is myopia, which is nearsightedness. So the focal point in front of the retina, which means that the eyeball is too long, and that's corrected with a concave lens. Okay, so you have a concave lens, which is going to allow you to focus the image at the focal point. Hyperopia is going to be a farsightedness, and that's the eyeball that's too short. So the focal point is behind the retina instead of on the focal point. Okay, this is corrected with a convex lens, so it refracts light less. And then you have astigmatism, which is unequal curvature in different parts of the cornea or the lens, and that's corrected with a cylindrically ground lens or laser procedures. There's other options here as well, but that is something that a lot of the ophthalmists use, as well as PTs, chiropractors will also have different tools at their disposal to correct this. Okay, so here's the normal eye. Light is refracted from the cornea, then again on both parts of the lens, and it focuses on the retina. Okay. Now, when you have nearsightedness, the eyeball is too long, the focal point is in front of the retina, and then the light diverges again, so you get blurry images. Okay, so that's uncorrected. Now you get a concave lens. Now you're going to diverge that light before it gets to the cornea, then it converges and converges again, so that now you get the proper convergence on the retina, so the eyeball is not too long anymore. So essentially you're going to diverge it so that enough so that your eyeball is going to correct it. And there's different intensities to that concavity. So the longer the eyeball, the more concave you need to make that lens that sits in front of the cornea. Okay. Now, if you have a hyperopic eye, which is for farsightedness, the eyeball is too short. Well, now that the image is too far behind the retina, so you get a blurry image when it lands on the retina itself. Now you get a convex lens, which looks exactly like the lens that you have in your eye, and you can increase the convexity in order to increase the convergence of the light rays. So it will bend the light in front of the cornea, bend the light again in the cornea, and bend the light on the lens so that you focus directly on the retina and get a clear image. Okay. So the functional anatomy of the photoreceptors is something that we're going to be discussing when we finish up the last portion of the lecture. So we're going to end this portion here, and we'll continue on with the last lecture from this from the online version, and then we will discuss more after exam 4.